Hi guys, welcome back to my channel. Today I am really excited to start a new biology playlist because I used to be a bio TA a couple years ago in my undergraduate. I really enjoyed the job because I got to break down complicated concepts into really simple language for my students, like what I'm trying to do now for this channel. And I remember my professor really gave the TAs great autonomy by letting us lead the discussion sessions all by ourselves without the professor even showing up. And I was leading 2 sessions per week, acting like a real teacher, lecturing 50 plus students, and plus the office hours and email communications. I really taught biology and know the stuff like today's topic, DNA replication, to my heart. And it was also the intro bio class that encouraged me to make the initial step of switching from pre-nursing to pre-med. All right, so excuse my elaboration. Usually I like to jump right into the topic, but I just got excited today starting this biology playlist. But without more further ado, let's talk about DNA replication. So as we know, DNA has this double helix structure, right? They're double-stranded. So before we can really do any sort of replication, we have to open up the DNA double strand, right? So the enzyme that does the job is called helicase, right? So helicase opens up double helix. Okay, all right. So the helix would open the replication bubble in 2 different directions. Okay, all right, and then helicase is not just doing the job all alone. He actually has 2 helpers, okay? So one being the single-strand binding proteins, okay? So single-strand and binding proteins are just proteins that bind it to DNA, the separated DNA strands, so that it prevents them from getting back together. Okay, so it prevents them from re-annealing. Okay, and then another one is topoisomerase, which is also called DNA gyrase on MCAT. I mean, I've never seen the word DNA gyrase on my biology class, but it shows up on MCAT. I just want you guys to know it so that you don't stress out too much when you see the new terminology. Okay, so topoisomerase, on the other hand, they kind of they're located here. Okay, so their purpose is to relieve the supercoil that's caused by the opening up of the double helix, okay? Because once you open it more and more, and then the 2 ends just get tighter and tighter. So topoisomerase, like, it's a very interesting mechanism. They literally like break the DNA bond and then reform it. So that's how they relieve the supercoil stress. All right. So that's the helpers of helicase. So now we have this 3 enzymes that's all working together to open up the DNA, so we can start the replication now. Okay. So before we start the details of the replication process, let's just differentiate the formation of a replication bubble in eukaryotes versus in prokaryotes. Okay, so I actually found a very good picture online showing that in eukaryotes we have this linear DNA structure, so we can have a multiple replication bubble that get formed at the same time, having multiple origins of a replication, right? But it's really not just about the structure of DNA being linear so that we can have multiple replication bubbles. It's more to expedite the replication rate so that multiple replication bubbles get formed together at the same time. Whereas in prokaryotes, we have this circular DNA, so only one replication bubble is formed, only one origin of replication, and then it's a circle. It goes around. So eventually you start from the origin of the replication, you go both ways, and then eventually you finish the replication after you go around the circle, right? All right. So that is the DNA replication in eukaryotes versus in prokaryotes. Okay. All right, so now let's start the process of DNA replication. Okay, so we always talk about directionality in biology class. You always hear the bio teacher say, okay, DNA synthesized goes from 5 to 3, right? So back in the days, you haven't taken organic chemistry. You actually don't really understand why does it have to go from 5 to 3, right? You haven't learned all the nucleophilic attraction, those things yet. So, you just, okay, remember, okay, 5 is a 5 prime carbon of the, which carbon? Sugar carbon of the DNA, right? Okay, so really, to decide which one is the leading strand, which one is the lagging strand, we have to tell the directionality, okay? So let's say if I have a 5 here and then 3 here, and then mentally you have to label the bubble 3 being here, 5 being here, and then 3 being here, 5 being here, and then 3 being here. Just rotate, just alternate the numbers, 5, 3, 5, 3, 5, 3, okay? And the other strand starts with 3, so 5, 3, 5, 3, and 5, okay? So I just alternate the numbers, and it's not a big deal. It's really not complicated, okay? So, and then we once again, we heard that, okay, we learned that we synthesize DNA from 5 to 3. Okay, so let me find my origin of replication. This is the origin, right? OOR, where I start the replication. So let's start 5 to 3, 5 to 3. Perfect. It's a continuous strand. This is a leading strand that goes from 5 to 3, right? Where's the other origin? The other origin is here. Okay, so which one goes from 5 to 3 continuously? This one. Okay, so this is the leading strand. Okay, and then the other strand, the origin is 3 end. But do we start from 3 end? No, it still goes from 5 to 3. Okay, it still goes from 5 to 3. But how? We just have to go backwards, right? Okay, we'll just use a different color. 5 to 3, 5 to 3, 5 to 3. But the difference is we don't get to really just synthesize it continuously, right? We just go as the bubble opens. The bubble opens a little bit, then we do this fragment, and then it opens more, we keep going, opens more, and then it opens more later, and then we keep going this way, right? From 5 to 3. Okay, so these are called fragments, right? Okazaki fragments. So Okazaki fragments is the lagging strand. Okay, so on the other DNA strand, we can see, okay, we have to go from 5 to 3, 5 to 3, 5 to 3. And as the DNA opens more, that we can have more 5 to 3 fragments, okay, fragments. All right, so that's how you tell which one is a leading strand, which one is a lagging strand, right? It's really simple. The leading one goes from 5 to 3. The lagging one still goes from 5 to 3, but in fragments, right? Okazaki fragments. Okay, so now we need to discuss about the primase, right? Because before we can continue the elongation of the DNA replication, we need to have some primer. Let me use a pretty, we have to have primer. And then each fragment, we need primer. Right? Primer has to be synthesized before the elongation of the DNA, okay? And then this is done by the enzyme primase, okay? So primase synthesize the primer, and then the primer is actually RNA, okay? It will later be ditched and then be replaced by DNA. But to start with, to begin this process, we have to have the RNA primers, okay? Whereas so I thought like in CPR it's also RNA primers, but actually in CPR it's in the lab, it's vitro, so it's a DNA primer, okay? All right, but in this biological scenario, it's a RNA primer, okay? So primase synthesizes RNA primer, okay? And then once the primer is there, the DNA polymerase can start its job of adding new nucleotides to the DNA strand to facilitate the replication, okay? And then also here, I noted that the DNA polymerase also has this proofreading function, okay? That prevents mistakes and mutation, okay? All right, so there's another enzyme we need to discuss, DNA ligase. What is DNA ligase, right? As you can see, the Okazaki fragments, once I got rid of those primers, right? This primer needs to be gotten rid of as well. So once I got rid of those primers, I have this nick, the little space between the fragments that needs to be sealed together. That's done by DNA ligase, okay? All right, so now that's all the enzymes that we have went over. So they seal the nicks. Okay. All right, so that's all the enzymes you need to know for MCAT. And then also you want to be able to differentiate which one is leading strand, which one is lagging strand. So for example, I give you this problem that, okay, please label if the upper strand or lower strand is the leading strand. Which one would you choose? Okay, and then a lot of students start to get confused because you're like, oh, look what I learned. I have 5 on the top, and then this is my origin of replication. I have my leading strand here. So voila, this is my leading strand. Is that correct? So once again, because DNA, it's a 3D structure, right? It's 3D in your body as well. It can really flip, it can really, I can change the orientation. So you can't just memorize the graph that you learned. You have to label 5 to 3, okay? So in this case, I have a 5 here. And then I will color label this. This is the 5 strand. And then this is my 3 end, okay? So to label this will be 3, this will be 5, and then for the red one, this will be 5, this will be 3. Oh, sorry, I just confused myself. So let's do the red one first. So the red one, we have 5 here, so we need to have 3 here, and then 5 here, okay? And then now let's do the yellow one. So the yellow one has 3 here, we have 5 here, and then 3 here. So we know the leading strand goes from 5 to 3, okay? So basically, this is the directionality going from 5, 3, the lower one being the leading one, okay? So if I flip the DNA again, would you be able to instantly tell which one is 5, which one is 3? So it's really not hard, just label the directionality, okay? All right, so now let's talk about why does it have to be synthesized from 5 to 3, okay? So like here, you see I've labeled the number of the carbon, right? Carbon number 1, that's attached to a base. Carbon number 2, 3, that has a hydroxyl group here. And then 4 and 5, that's attached to a phosphate. Okay, so the 3-end carbon has a hydroxyl, okay, here. So it has a hydroxyl that really, this proton can be pronated by some sort of base in the solution, okay? And then once it gets depronated, this oxygen become a negative nucleophile, okay, which is electron dense. Okay, and now where's my electrophile to be attacked, right? So as we can see, we have this material of a DNA replication called a what's it called? It's called DNTPS, right? They're called deoxyribonucleoside triphosphates. So they're the building material, the nucleotides. I mean, they have triphosphates, not one phosphate yet, okay, to be a real nucleotide that gets attached to DNA. But as the building material, they have 3 phosphates, and then my phosphorus here, this P here, is actually partial positive. Why? Because I actually have these negative oxygens covalently. They're actually called a coordinate covalent bond, meaning that only the oxygen is contributing the 2 electrons between the bond instead of each atom contribute 1 electron. That's the normal covalent bond, right? So in this case, whatever this special covalent bond is, they kind of draw away the negative charge of the oxygen, right? And then also it's a stabilization of this negative thing by the positive cofactor magnesium 2+, right? Okay, so all the reason combined that makes my phosphorus positive, partial positive, okay? Which makes it what? A good electrophile, okay? So now it's perfect to be attacked by my oxygen, okay? So oxygen attacks it and it forms a OP bond, right? So if I were to label this on the graph, here's the OP bond, there's the OP bond, that's formed, okay? So when the OP bond is formed, a leaving group has to leave, right? You form bond, you break a bond, and then the diphosphates are the leaving group that leaves, okay? So this part. So you break this OP bond, okay? So you form the phosphodiester bond, okay? So this is how you must go from 5 to 3, okay? Because if you look at the sugar that has been formed, not sugar, I mean the nucleotide. So this is 5 here, and then 1, 2, 3, 4, 5. Okay, 3 is the end. So you look at the whole picture, the nucleic acid, you see that 5, we go from 5 to 3. Okay, that's really the number of the carbon of the sugar of DNA. Okay, and then once again, the 3 end has this hydroxyl group that acts like a nucleophile that attacks the phosphate group, right? So that's why we always like this 3 end is really important, okay? It's the attachment point. All right, so I hope that's helpful. Comment down below if you have any questions or if you think this makes sense. And then if you are just as helpful as I would like to be, you can also put your answer in the comments regarding these two scenarios, which one is the leading strand. I hope that's helpful.